Hello, good evening, all of you. Hope you are all doing fine. Am I audible? Can you hear me correctly? Abhinav? Can you hear me? Yes. Do you have any doubt on the topic we were discussing last time? I was telling you about the mechanism of inspiration and expiration. So remember, inspiration occurs because of the contraction of the muscles. That is why inspiration is called active. Whereas expiration occurs when these muscles are relaxing, no work is done, no energy is required. So expiration is passive. 1 point. Number 2, inspiration occurs when the pressure inside the lung is decreased. That decrease in, fall in pressure is called negative pressure. So when the pressure of the lung falls below very much, there is a gradient between the pressure in the atmosphere and pressure inside the lung. So because of that gradients, air enter in, inspiration occur. And when pressure inside the lung increases, how pressure inside the lung is increasing, volume of the thoracic cavity decreases. When the thoracic cavity decreases in volume, pressure inside the lung is increased, it becomes higher than that of the atmosphere, air goes out. That is expiration. Any doubt? Anybody, Makala? And for making expiration also active, you are having two sets of muscles. They are called expiratory muscles. What are they? Internal intercostal muscles. Muscles which are all present in between the ribs on the inner side. That is called internal intercostal muscle. External intercostal muscle is inspiratory muscle. This is internal intercostal muscle, which is expiratory muscle. Then muscles in the abdomen. So using the muscles of the abdomen, you can make okay. So now you are having all these topics coming. Anybody having any doubt? Anybody? NCERT book വന്നിട്ടില്ല. Anybody having doubt? അപ്പോൾ we can make expiration also active. Then it is called labored breathing. അപ്പോഴാണ് നമ്മൾ അതിനെ labored breathing. Okay. Anybody having a doubt on that? Okay. We have the ability to increase the strength of inspiration and expression with the help of additional muscles in the abdomen. That's an important point you have to remember. Using muscles in the abdomen, additional muscles, we can make both inspiration and expression more powerful. On an average, we breathe 12 to 16 times. When you are all working, rate of breathing will increase. When you are in perfect rest, as if you are in sleep, it will be minimum. So it varies from 12 to 16 times a minute. Volume of air involved in breathing movement can be estimated by a small instrument called spirometer. Using a small instrument called spirometer, you can measure the volume of air you are taking in and giving out. So, volume of air involved in breathing movements. And why we are all measuring it? Because these values are helping in assessing the pulmonary functions. When you are consulting a pulmonologist, when you are having a problem in breathing, then he will give you this instrument to which you will be asked to breathe and the values will be measured. Okay. Now, what is that instrument? I will give you a brief account on that. This is a gauged meter. Okay. You will be having meter. That is why I told you it is a gauged meter. And there will be a ball inside this. This ball will be moving along this gauge meter so that the pulmonologist can evaluate the amount of air that you will be taking in and breathing out. This is connected to a rubber tubing. Okay. Here you are all asked to keep your mouth, and through the mouth you are all breathing in, breathing out. Okay. Not through the nostril in the instrument. So, normally you are all taking air through the nose. Now you can breathe it with your mouth. It is only for a short period, no problem. Okay. So, depending upon the amount of air that you are all pushing, the ball will be moving up and down. So, the value can be measured. This is the spirometer. Simple. Okay. And using this spirometer, you can measure certain values which are called volumes. So, volumes are the values you can measure directly from spirometer. Okay. What are these volumes? Now, I will be giving you a graph which all of you should take down. These lines are marked as A, B, C, D and E. Okay. Now, normally when you are all breathing, you take in same amount of air and give out same amount of air. So, amount of air that you are all taking in is also equal to amount of air that you are all giving out. When you are all normally breathing, peacefully breathing at rest. So, this is marked in graph B to C like this. Okay. This is called tidal volume. What is that value? Volume of air, normally a man breathing in is equal to volume of air he is breathing out. This is called tidal volume, volume of air that is entering in and giving out normally in a normal breathing. It is 500 ml. So half a liter normally we are taking. But over and above, you can take additional volume. For example, I have breathed in air. Okay, so this is my inhaling. Over and above, I can take. Okay, all of you can do it. That volume of air we are taking after a normal inspiration. After a normal inspiration. So, this is what we are all inspiring from A to B. This is called inspiratory reserve volume, additional volume, reserve volume you are all taking in addition to normal in breathing. It is called inspiratory reserve volume A to B. This is usually 2500 to 3000 ml. So almost 3 liters of air you can take. This is called inspiratory reserve volume. That is additional volume you can inspire. That's the meaning. Similarly, you are all giving out air, expiration, and also you can give out additional volume of air. After a normal expiration, you can give some more amount of air from your lungs. This C to D, additional volume of air you are all sending out is called expiratory reserve volume. Additional volume you are all expiring. After a normal expiration, what is? That is from C to D. This ERV is usually 1 liter to 1.1 liter, 1100 ml. Whatever may be the force you are all using, you cannot send all the air. Don't think that you have already sent all the air in your lung during expiratory reserve volume. No. Nobody can send all the air that is present inside the lung after a normal expiration. See, after a normal expiration, still large amount of air is remaining in your lungs. That include air you can send out and air you cannot send out. There is still some amount of air that nobody can send out. This is called residual volume, which is 1100 to 1200 ml. Now, the doctor will be asking you, to take maximum amount of air and then give out air. So you have taken maximum. So normal inspiration plus additional inspiration. So you have taken maximum amount of air. Okay. Then give out maximum. This will be the additional volume you have taken in, normal volume you have taken in, and additional volume of air you can send out. So from A to C, A to D, that will be the total volume you can take in after a normal or after a expulsion, compulsory expulsion or maximum expulsion. Okay, or that is also equal to total amount of air that you can send out after a forceful inspiration. So normally doctors will be measuring these. Okay, so 4 volumes you are getting directly. Okay. Actually, RV, reserve volume, you cannot measure directly from the spirometer. The values you are obtaining are IRV, TV and ERV. So, volumes are values that obtain from spirometer directly. Okay. Now, by adding 2 or more values, രണ്ടോ അതിലധികമോ വോള്യൂമുകളെ കൂട്ടിച്ചേർത്തുകൊണ്ട് you calculate certain values which are called capacities. They are called capacities. Capacities are not directly measured from spirometer, but we are all calculating. What is that? This is the volume that you are all additionally taking. This is the volume that normally you are taking. So normally taking plus additional taking. These two together is called inspiratory capacity. That is the capacity, maximum volume of air you can inspire, take in after a normal expiration. After a normal expiration. So what is its value? It is 3000 to 3500 ml. Similarly, this is the volume one can breathe out, expire. This is the volume one can breathe out in additional. So, from B to D is the total volume of air one can send out after a normal inspiration. After a normal inspiration. This is called expiratory capacity. So what is its value? 1500 to 1600. You have still some amount of air remaining in the lungs after a normal expiration. സാധാരണ ബ്രീത്തിങ് കഴിഞ്ഞാലും ലങ്ങിനകത്ത് എത്ര എയർ ഉണ്ട്? Total amount of air that is remaining in your lungs after a normal expiration. That include volume of air you can send out additionally, that is ERV, plus volume of air you cannot send out, that is RV. This is called functional residual capacity, FRC. അപ്പോൾ എത്രയാണ് അതിൻ്റെ വാല്യൂ? It is 2100 to 2100 to 2300. Can you follow me? മനസ്സിലാവുന്നുണ്ടോ മക്കളെ? യെസ്. എപ്പോഴും ചോദ്യങ്ങൾ ചോദിച്ചും എപ്പോഴും എൻ്റെ കൂടെ ഉണ്ടാവണം കേട്ടോ. വെരി ഗുഡ്. അപ്പോൾ here you can send out plus here you cannot send out. ഈ രണ്ടും കൂടി ചേർന്നതാണ് air that is remaining in the lung after a normal expiration. It is called FRC, functional residual capacity. ഇനി after a deep in-breathing, inspiration, what is the total volume of air you can send out? That is from A to D. That is called vital capacity. That is volume of air you can send out after a deep inspiration. Strong inspiration ശേഷം എത്ര എയർ മാക്സിമം കളയാൻ പറ്റും? അതായത് that is additional volume you have taken, you will be sending out. Normal volume of air you will be sending out during a breathing, plus additional volume of air you can send out by forcible expiration. These three together is called vital capacity. So what is its value? 4000 to 4600. 3.5, 4.5 plus 1. So 4.6 liters. That is called vital capacity. Vital capacity can be also defined in another way. What is the total volume of air you can breathe in after a forceful expiration? After a forceful expiration, what is the total amount of air you can breathe in? അപ്പോൾ അഡീഷണൽ ആയിട്ട് കളഞ്ഞത്, സാധാരണ എടുക്കുന്നത്, അഡീഷണൽ ആയിട്ട് എടുക്കാൻ സാധിക്കുന്നത് അതൊക്കെ നിങ്ങൾ ഉള്ളിലേക്ക് എടുക്കും. ദാറ്റ് ഈസ് ആൾസോ ഈക്വൽ ടു ടോട്ടൽ വോളിയം ഓഫ് എയർ യു ആർ ആൾ ബ്രീത്തിങ് ഔട്ട് ആഫ്റ്റർ എ ഫോഴ്സ്ഫുൾ ഇൻസ്പിറേഷൻ. അപ്പോൾ രണ്ട് തരത്തിലും വൈറ്റൽ കപ്പാസിറ്റിക്ക് ഡെഫിനേഷൻ കൊടുക്കാം. റിപ്പീറ്റ് ചെയ്ത് പറഞ്ഞുതരാം. Vital capacity is the total volume of air one can breathe out, blow out after a maximum intake, maximum in breathing. It is also equal to volume of air you can take in after a forceful expiration. This vital capacity doctors will examine. Okay, still there is some amount of air we have not considered, that is residual volume. So vital capacity plus that residual volume is called total lung capacity. So what will be its value? It is 5100 to 5800. So almost 6 liters of air. That is the total amount of air our respiratory system may be containing. Is that clear? The graph will be helping you a lot, as questions will be coming. to arrange these volumes or values in an ascending order or a descending order. Arrange the volumes, pulmonary volumes in an ascending order. Questions may be coming like that. Or in a descending order. Ascending barna smaller first, larger last. Descending na first the larger volumes, then smaller volumes. Okay. Or capacities. Ascending order, descending order. Now all the values are here. So understand of which is the largest one? Total lung capacity. So please write what will be your answer if the capacities are arranged in a ascending order, ascending. Smallest one, expiratory capacity. Then you are having functional residual capacity greater than is inspiratory capacity, IC. Then you are having vital capacity, and the greatest one is total lung capacity. Is that clear? Vital capacity എന്ന് പറയുമ്പോൾ രണ്ട് വിധത്തിൽ ഡിഫൈൻ ചെയ്യാം. Now, what is the total volume of air you can blow to that machine, spirometer, after a maximum intake? Okay. After a maximum intake, എത്രത്തോളം നമ്മൾ ബ്ലോ ചെയ്യുന്നു അതാണ്. അല്ലെങ്കിൽ ഇത് തന്നെയാണ് ആഫ്റ്റർ maximum blowout. What is the total amount of air you are all taking in? എത്രത്തോളം നമുക്ക് എടുക്കാൻ പറ്റും? എടുക്കുന്നതിനെ അളക്കാൻ ബുദ്ധിമുട്ടാണല്ലോ. പുറത്തേക്ക് കളയുന്നതിനാണല്ലോ ട്യൂബിലേക്ക് ഊതി നമുക്ക് കിടന്നത്. അപ്പോൾ അതാണല്ലോ എളുപ്പം മെഷർ ചെയ്യാനായിട്ട്. രണ്ടും ഒന്ന് തന്നെയാണ്. Total volume of air you can blow out after a maximum intake is also equal to maximum amount of air you are all taking in after a forceful expiration. This includes IRV plus TV plus ERV, or it is also equal to TLC minus RV. പല ഡെഫിനിഷനും കൊടുക്കാൻ പറ്റും. ഇറ്റ് ഈസ് ആൾസോ ഈക്വൽ ടു IRV plus TV. This is the vital capacity. IRV plus TV is called IC. So it is also IC plus ERV. Correct? Now, ERV plus TV is EC. So it is EC plus IRV. So there are so many definitions for vital capacity. That is why you should have this graph. Then only it will be more clear to you. Okay, Makle? Is it clear, this parameter? So why these values are important? Because they are all very important in clinical assessment of your lungs. In clinical examination, pulmonologists will check these values. Clinical assessment of pulmonary functions. Now you are having these values. Please read that part. Vital capacity is it now clear, Makle? Vital capacity and total lung capacity is equal to vital capacity plus residual volume. Vital capacity plus residual volume. Any doubt? Okay. So after finishing the first part, that is breathing in, breathing out. We go to the second stage in breathing. What is that? Exchange of gases. So you have taken air into the lungs. Now it is in the alveoli. How the gases are exchanged? Remember, gas exchange takes place only by a simple mechanism called diffusion. No other mechanism. Gas exchange is always taking place through a very simple mechanism, diffusion. Diffusion is a passive mechanism. No energy is required. Molecules will move from a region of high partial pressure to a region of low partial pressure. So, for diffusion to take place, there must be a gradient of pressure. What is needed is simply we need a gradient. There must be a difference from a higher level to a lower level. Only gases or fluids will be diffusing. Okay, so the exchange of gases both at alveoli, that is the primary exchange, and secondary exchange at tissue level is because of these gradients, no other mechanism. Let us see how our air is reaching our tissues. Air is a mixture of gases and it consists of mainly nitrogen, almost 79%, oxygen, about less than 21%, and small amount of minor gases. So it's a mixture of gases. And the pressure exerted by only one component, one gas in this mixture of air is called its partial pressure. We write this partial pressure as P. O2. So, how you calculate the partial pressure of oxygen in air? Air is having pressure of 760 at normal mean sea level, and it is almost 20%. You just calculate 20 by 100. This is the percentage. So 20 by 100. Then you get about 159. Because it is almost 20.8, almost 21. So we roughly calculate. Now you got an idea. So we are having 20.8 roughly. So atmospheric air will be having oxygen at the pressure, partial pressure of 159. Similarly, what is the partial pressure of carbon dioxide? PCO2 760 into, we are having only 0.04 by 100. 0.04%. So it is just 0.3. That is the partial pressure of oxygen. So mm of mercury. Okay, this is the partial pressure of oxygen and carbon dioxide, which are the gases involved in our breathing. Okay, so this air you are all taking into lungs. Remember how much air you are taking? Only 500 ml per breathing. Already in your lungs there is foul air, bad air, which is the residual volume. Also, you are having air that you can send out. That is, expiratory reserve volume is also there. So, you are having a total functional residual capacity more than 2 liters. You are taking only 500 ml. More than 2 liters are there already in the lungs, and that has been remaining there for some time, which is rich in carbon dioxide. So what happens to, understand this point very carefully, what happens to the percentage of oxygen of air that is reaching the alveoli? It decreases. In atmosphere, percentage is 20.8, but in alveoli, once it mixes, half a liter mixed with more than 2 liters of bad foul air. So oxygen percentage is decreasing to about 16, average 16, and carbon dioxide to almost 4. Okay, so because of this change, or because of this mixing of fresh air with foul air or bad air, air that is rich in carbon dioxide inside the lung, there is a change in the percentage of oxygen and carbon dioxide. Because of this reason, the pressure of oxygen, partial pressure of oxygen in the alveolar air is reduced to 104. Now you got the idea? It is reduced to 104. This is the reason. In atmosphere, partial pressure of oxygen was 159, but on reaching the alveoli, it is reduced to 104. And what happens to the partial pressure of carbon dioxide? It is increasing to 40, almost 100 times, more than 100 times its partial pressure is increasing. So oxygen partial pressure decrease, carbon dioxide partial pressure increase. This is because that the volume of air we are taking in, fresh air we are taking in during inspiration is only half a liter, and it is mixing with more than 2 liters of air that are already present inside the lung. Okay. So this is the partial pressure of oxygen 104 and that of carbon dioxide 40. Now, this air atmosphere, oxygen is 159. On reaching alveoli, it becomes 104. Now, you are having blood coming to the alveoli. What type of air? Impure blood or venous blood. So, we call it as venous blood. It is the venous blood. Venous blood is amount of oxygen will be very, very less. It is as low as 40. Why it is so, we'll be discussing. So here the oxygen level is as low as 40 because most of the oxygen was given to the tissues. So now it is containing only 40. So in alveoli 104, in blood 40. So there is a deep gradient. Always oxygen will diffuse from a region of high concentration to a region of low concentration. Okay, so this is the reason why excuse me, oxygen is diffusing from alveoli to the blood. Now, when oxygen reaches the venous blood, the concentration in blood gradually increases. Oxygen level is increasing. Similarly, carbon dioxide is diffusing out. So, carbon dioxide is diffusing from blood to the alveoli, oxygen is diffusing in. So, as a result of that, blood become oxygenated. We call it as oxygenated blood. So, venous blood become arterial blood. Generally, it is oxygenated blood. Okay. So, oxygen rich oxygenated blood will be having oxygen at a partial pressure of 95. So, why oxygen diffused? Because of that gradients. Now, this blood is reaching the tissues. When it reaches the tissues, tissues are the site where oxygen is utilized by the cells, whereby energy is released. So oxygen is completely or quickly utilized so that level of oxygen in the tissues are very low. So what is the amount of oxygen in the blood? 95. The pressure of oxygen is only 40. Why oxygen is very low? Because oxygen is continuously utilized. It is taken up by the cells. So oxygen partial pressure is very low. So oxygen from the oxygenated blood will be diffusing into the cells. Okay. So here also secondary exchange is also taking place because of this gradients in pressure, that is by simple diffusion. So from 95 to 40. Is that clear? So 104 to 40, then it increases in blood to 95. That 95 oxygenated blood is reaching the tissues. It is diffusing to the cells because in cells the pressure is very low. Now, what about the carbon dioxide? Carbon dioxide is produced in the cells. Its concentration is 45. Its concentration is 45. And what is the partial pressure of blood, that is oxygenated blood, reaching the tissues? It is just 40. Partial pressure of carbon dioxide in oxygenated blood reaching the tissues will be 40. So here also there's a gradient. Then you may ask, only 5 mm of mercury, that is the difference. Oxygen 104, 40. ഇവിടെ 45, 40. അതെങ്ങനെയാണ്? ഇത്ര ചെറിയൊരു പാർഷ്യൽ ഡിഫറൻസ് മതിയോ കാർബൺ ഡയോക്സൈഡ് ഡിഫ്യൂസ് ചെയ്യാനുള്ളൊരു സംശയം ഉണ്ടാവും. ഓക്സിജന് വലിയ ഡിഫറൻസ് ആണ് അല്ലേ? ഡിഫറൻസ് ഇൻ partial pressure, gradients in partial pressure was very high for oxygen, but for carbon dioxide only small. But for carbon dioxide it is sufficient. Why? It is because that carbon dioxide is having 20 to 25 times more solubility. Carbon dioxide is having 20 to 25 times more solubility than oxygen. Oxygen is having double the solubility than nitrogen. That is why, though nitrogen is present in large amount, oxygen is quickly diffusing because its solubility is more. Remember, in diffusion of gases, I told you the most important point is gradients. Only from a region of high partial pressure to a region of low partial pressure, gases are exchanged, diffusing. That is gradients in pressure. Number 1. 2. Solubility. Number 2 factor is solubility. Greater the solubility, greater is the diffusion capacity. So carbon dioxide is having 20 to 25 times more solubility. So its diffusion capacity is 20 to 25 times more than that of oxygen. So a difference in small 5 mm of mercury is sufficient for carbon dioxide diffuse. And third one is the diffusion membrane. What is its thickness? Remember, we are having the diffusion membrane. What is the diffusion membrane? The membrane or the layer that separates alveoli and the blood. It is actually made up of 3 layers of cells. What are they? On the innermost one, you are having a wall of alveoli, which is made up of squamous epithelium, thin, flat, squamous epithelium. thin, flat, squamous epithelium means cells which are all thin, tile-like, very thin, that will be allowing exchange of gases. That is the innermost. Then you are also having the blood vessel wall, which is having the endothelium of blood capillary. Capillary is made up of only one layer of cells, which is also flat squamous epithelium. That layer is called endothelium. And these two epithelia are connected together by, these two are all connected together by a jelly-like. Okay. You are having a basement substance. It is not a cellular layer, but a matrix. This matrix is called the basement substance. So, actually there are three layers. What are they? One, squamous epithelium of the alveolar wall. Two, the basement substance. And three, the endothelium of the blood capillary. So, oxygen is diffusing across these three layers. As it diffuses from alveolar cavity to the blood. Though there are three layers, the total thickness is less than 1 millimeter. Alveoli are all very thin walled, very thin walled. Though you are having three layers, what are they in the order? Squamous epithelium of the alveolar wall, basement substance, endothelium of the blood capillary of the alveoli. The total thickness is less than 1 millimeter. So, this acts as the diffusion membrane, the membrane across which diffusion is occurring. That is called diffusion membrane. So, third important factor is thickness of the diffusion membrane. So, what are the three factors? I will repeat once again. First one, the gradients. That's the most important one. Two, solubility, diffusion capacity. Three, the thickness of the diffusion membrane. So, all these three factors are favoring the easy diffusion of gases. Okay. This is the reason why the oxygen is diffusing from alveolar cavity into the RBC. Same reason. You are having capillaries in tissues. So, they are all crossing to the cells across the endothelium, only endothelium. That endothelium is very thin. Endothelium is the only wall present in the blood vessel. It is very, very thin. So, it will easily cross the endothelium and reach the cells. Thus, oxygen reaches the cells. Similarly, carbon dioxide from cells diffuse back into the blood. So, what happens to the level of carbon dioxide? As more and more carbon dioxide diffusing, the level of carbon dioxide increases to 45. Now, what's the nature of that blood? Nature of the blood is now deoxygenated, which we simply call it as venous. Okay. Carbon dioxide from tissues diffuse into the blood vessel, so that carbon dioxide partial pressure increases in the blood from 40 to 45. Now it is called venous blood. So arterial blood or oxygenated blood become deoxygenated because oxygen is removed. Remember, though we say it is deoxygenated blood, not entire oxygen is removed. Blood is giving only about about 25% of the oxygen to the tissues. 75% is retained within. I will tell you about that later on. So now blood is becoming deoxygenated, which is having only 45 partial pressure carbon dioxide. So oxygen is diffusing, and as oxygen is diffusing, its partial pressure decreases to 40. So carbon dioxide partial pressure increased to 45, oxygen partial pressure decreased to 40. So it is that type of blood that is coming. So oxygen partial pressure PO2 40, partial pressure of carbon dioxide PCO2 45. This is the situation in the venous blood or deoxygenated blood. That is reaching again alveoli. Remember, what is the partial pressure of carbon dioxide? 45. And what is the partial pressure of carbon dioxide in alveoli? It is just 40. So from 45 to 40, carbon dioxide will be easily diffusing out. So both in the tissues as well as in the alveoli, carbon dioxide is diffusing because of the gradients in pressure. Similarly, oxygen is diffusing from alveoli to the blood and from blood to the tissues also because of that diffusion, simple mechanism of diffusion, no other mechanism. And it is by maintaining a gradients of pressure gradients. That is the only mechanism that favor this diffusion. So blood become oxygenated, and in oxygenated blood you will be having the partial pressure of oxygen 95 and partial pressure of carbon dioxide is decreasing to 40. So in oxygenated, partial pressure of carbon dioxide is 40, partial pressure of oxygen is 95. In venous blood, partial pressure of oxygen is 40, partial pressure of carbon dioxide is 45. So more carbon dioxide is present than oxygen. That is why it is called deoxygenated. In oxygenated, we are having more partial pressure for oxygen 95. Partial pressure of carbon dioxide is 40. Is it clear now? So no other mechanism. Yes, thickness is total thickness of the wall of the alveoli, wall of the diffusion membrane. That is the separation between gas in the alveoli and blood in the blood vessel. That is constituted by 3 layers. Okay. Any other doubt? Anybody, Makale? Others? Abhinav? Can we go to the next part? Namely, transport of gases. So, oxygen is to be transported from alveoli to tissues, and carbon dioxide is transported from tissues back to alveoli. So, blood acts as the transporting medium. Okay. About 97 of oxygen is transported. We will be now going to the transportation of oxygen. Let us study how oxygen is transported. Oxygen is transported in two ways. One, oxygen is a gas that is sparingly soluble in water. We will be studying that in blood you are having 55% plasma, that is the liquid part, and of that 90 to 92% is water. So blood contains a lot of water. That's why blood is a fluid tissue. And in that water, oxygen may be dissolving. Oxygen is not a good soluble gas. It is sparingly soluble. So 3% of the total volume of oxygen is dissolving in plasma. So it is, what is happening in a physical form. 3% of oxygen is getting dissolved. I told you solubility of oxygen is double than that of nitrogen. So it is dissolving in plasma of water. Thus plasma carries them in a dissolved form, in a physical form. The remaining 97% is carried in a chemical form. It is transported in a chemical form. How? Oxygen combined with a complex protein called hemoglobin reversibly to form oxyhemoglobin. To form a chemical compound called oxyhemoglobin. Hemoglobin is not oxidated. It is just oxygenated. Oxygen is added. Okay. Where is that hemoglobin? What is that hemoglobin? Hemoglobin is present in RBC. Hemoglobin is a pigment. That is why our blood cell is red. And those which are having hemoglobin is called red blood cell, RBC. So inside that we are having hemoglobin. And what is that hemoglobin? One molecule of hemoglobin, okay, one molecule of hemoglobin has heme plus globin. Globin is a protein. We are having hemoglobin with alpha chain and beta chain. Two alpha chains and two beta chains. Okay. Just understand the basic things. Highly complex alpha chain, and the other one is slightly different beta chain. So this is beta, this is alpha. So you are having 4 globin molecules. Okay, globin molecule will be having 4 peptide chains or amino acid chains. 2 alpha chain, 2 beta chain. Okay. Now each of them will be having a heme. So 4 heme plus 1 globin. That is the structure of one hemoglobin. That one globin is made up of 2 alpha, 2 beta chains, and each of them you have one heme. What is a heme? Heme will be containing one ion. They contain an ion in the center. In the ferrous form. Okay. So you are having 4 heme. Heme is containing, heme is a now you got an idea. Heme is actually a porphyrin having 4 pyrrole rings which are interconnected, and you are having ferrous ion. This is a heme. ഇതെല്ലാം മറന്നുവിട്. It is just for you to understand the basic things. Okay. You are having 4 heme plus 1 globin. That globin is having 4 peptide chains, 2 alpha chain, 2 beta chain. That is the nature of one molecule of hemoglobin. This hemoglobin will take one oxygen molecule, each with one ion of one heme. There are 4 heme, and thus it can take 4 oxygen with them. It will be combining with maximum 4 oxygen. ഓരോ ഹീമിനും ഓരോ ഓക്സിജൻ എടുക്കാനുള്ള കഴിവുണ്ട്. മനസ്സിലായോ? Each heme can take 1 oxygen. പക്ഷേ ഓക്സിജനുമായിട്ട് കമ്പൈൻ ചെയ്യണമെങ്കിൽ sufficient partial pressure should be there. If there is sufficient pressure, then only oxygen will be coming and combining. 1 will be coming, then 2 will be coming, 3 will be coming, then finally the last one. When all the 4 oxygen is taken, then we call it as saturated. Now, hemoglobin is said to be saturated. It cannot take more oxygen. And the partial pressure required for taking all the 4 oxygen with them by what hemoglobin is called saturation pressure. Okay, this is a very, very important part in our chapter. We are having saturation, which is depending upon partial pressure of oxygen. So there is a connection between partial pressure and saturation level, which we can draw in a simple form. This diagram is called shows the relationship between percentage of saturation and partial pressure of oxygen. If you plot the relation between partial pressure of oxygen with saturation, so for example, this is the 100% saturation, what you get is a sigmoid curve, an S-shaped curve. This is called oxygen dissociation curve. Not oxygen association. It is oxygen dissociation curve. എന്താണ് അങ്ങനെ പറയുന്നതെന്ന് ഇത് കഴിയുമ്പോൾ നിങ്ങൾക്ക് മനസ്സിലാവും. Only oxygen is sparingly soluble മോനെ. ഓക്സിജന് വളരെ കുറച്ചേ സോളബിലിറ്റി ഉള്ളൂ. ഹൈലി സോളബിൾ ആയിട്ടുള്ള ഒരു ഗ്യാസ് അല്ല. അപ്പോൾ വെള്ളത്തിൻ്റെ അളവും കുറവാണല്ലോ. ആകെയുള്ള ബ്ലഡിൻ്റെ 55% മാത്രമാണ് പ്ലാസ്മ. അതിൻ്റെ 90% ആണ്. അപ്പോൾ amount of oxygen that can be transported in a physical form is reduced. That is why only 3% is transported. ബാക്കി 97%ഉം in a chemical form ആണ്, ഈ രൂപത്തിലാണ്. When the partial pressure of oxygen is very low, percentage of saturation will be also low. When you increase the partial pressure, then saturation also increases. Now it reaches a maximum. ഈ മാക്സിമം 100% സാച്ചുറേറ്റഡ് ആയി കഴിഞ്ഞാൽ നോക്കുക, പിന്നീട് എത്ര നിങ്ങൾ പാർഷ്യൽ പ്രഷർ കൂട്ടിയത് കൊണ്ടും സാച്ചുറേഷൻ ഒന്നും കൂടില്ലല്ലോ. മാക്സിമം 4 ഓക്സിജനല്ലേ എടുക്കാൻ പറ്റുള്ളൂ. 4 മോളിക്യൂൾ ഓഫ് ഓക്സിജനാണ് ഒരു ഹീമോഗ്ലോബിൻ എടുക്കാൻ പറ്റുക. മാക്സിമം 4 മോളിക്യൂൾ. അത് ഏതാണ്ട് നമ്മുടെ അറ്റ്മോസ്ഫിയറിൽ അൽവിയോലിക്കകത്തുള്ള പ്രഷറിൽ നമ്മുടെ ഏതാണ്ട് 97% ഓക്സിജനും സാച്ചുറേറ്റ് ആവുന്ന വിധത്തിലാണ് ഉള്ളത്. ഓക്കെ, സോ അറ്റ് അബൌട്ട് 97 പാർഷ്യൽ പ്രഷർ നമ്മുടെ ബുക്കിലുണ്ട്. സി വി ആർ ഹാവിംഗ് ദാറ്റ്. ആൾമോസ്റ്റ് 90 കഴിഞ്ഞ് കുറച്ചുകൂടി വരണം കേട്ടോ. ആ 97 ആകുമ്പോഴേക്കും 100% സാച്ചുറേഷൻ ഈസ് ഒക്കറിങ്. വി ആർ ഹാവിങ് 104 അല്ലേ? 104 ഉണ്ട് അൽവിയോലിയിൽ. സോ oxygen will be fully mixing with the hemoglobin and hemoglobin is fully saturated. അപ്പോൾ 100% saturation ഏതാണ്ട് എത്തുന്നുണ്ട് പാർഷ്യൽ പ്രഷർ ഓഫ് ഓക്സിജൻ. അതിൽ കൂടുതലായിട്ടും കാര്യമില്ല. എന്താണ് കാരണം? ബിക്കോസ് ഇറ്റ് ഈസ് ഫുള്ളി സാച്ചുറേറ്റഡ്, നോ മോർ ഓക്സിജൻ ക്യാൻ ബി ടേക്കൺ. സോ ദിസ് ഈസ് 50%. ഇവിടെ ശ്രദ്ധിക്കേണ്ട ഒരു കാര്യം, വാട്ട് ഈസ് ദി partial pressure at which 50% saturation occur? അത് ബുക്കിലില്ലാത്തൊരു കാര്യമാണ് ഇപ്പോൾ പറയുന്നത് കേട്ടോ. ഇറ്റ് ഈസ് കാൾഡ് P50 വാല്യൂ. എന്താണത്? P50 വാല്യൂ. 50% saturation occur at about 27. പാർഷ്യൽ പ്രഷറിൽ 50% സാച്ചുറേഷൻ നടക്കും. ക്രമേണ partial pressure കൂടുമ്പോൾ 100% saturation നടക്കും. Okay. This is the relation between partial pressure and saturation, which is shown in the graph called oxygen dissociation curve. Okay. Oxygen dissociation curve. ഇതാണ് P50 വാല്യൂ. ഇതാണ് 100% saturation value. This curve is called oxygen dissociation curve and is highly useful in studying the effect of factors like partial pressure of carbon dioxide, hydrogen concentration, etc. on binding of oxygen with hemoglobin. Now, what is the situation in alveoli? In alveoli, what is the situation? Partial pressure of oxygen is high. Partial pressure of carbon dioxide is low. What is the temperature? Temperature is also low. And what is the hydrogen ion concentration? It is also low. Hydrogen ion concentration low means pH is high. pH high means alkaline condition. Blood is more alkaline. So these are all the factors in alveoli. And these factors are all helping in combination of oxygen with hemoglobin. So oxygen and hemoglobin will be easily binding together. So the reaction goes to the right, that is Hb plus O2 giving HbO2. That reaction goes to the right. Okay. Hemoglobin will be combined with the oxygen to form oxyhemoglobin. So all the factors in the alveoli are all favoring the combination of oxygen with hemoglobin. Whereas in tissues, the situation is entirely different. What are the conditions in tissues? Partial pressure of oxygen is low. Partial pressure of carbon dioxide high. Temperature, tissues are the region where metabolism is occurring. So, as a result of metabolism, heat is produced. So, heat is high, temperature is high. Hydrogen ion concentration is also very high. Hydrogen ion concentration high means pH is low. pH low means acidity. So the condition is just the reverse. So when the condition is just the reverse, the association between oxygen and hemoglobin become very much reduced. The main villain in reducing the affinity of oxygen with hemoglobin is carbon dioxide. So because of the higher concentration of carbon dioxide, this carbon dioxide will reduce the affinity of oxygen with hemoglobin. So oxyhemoglobin splits. ഓക്സിജനും ഹീമോഗ്ലോബിനും തമ്മിലുള്ള ബന്ധത്തിനെ കുറയ്ക്കുന്നവനാണ് കാർബൺ ഡൈ ഓക്സൈഡ്. കാർബൺ ഡൈ ഓക്സൈഡ് ടിഷ്യൂവിൽ ധാരാളമായിട്ടുണ്ട് താനും. അപ്പോൾ oxyhemoglobin ടിഷ്യൂവിലെത്തുമ്പോൾ you are having more carbon dioxide. This carbon dioxide will be causing the breaking of oxyhemoglobin. That effect is called, and ipo parayunnathu bookililla, Bohr effect. Please take down what now I am telling. Lowering of affinity of hemoglobin with oxygen by the high partial pressure of carbon dioxide in tissues, by the high partial pressure of carbon dioxide in tissues is called Bohr effect. It is called Bohr effect. So, when carbon dioxide is more, what happens? Will the hemoglobin 100% saturated? No. It become reduced. It become reduced. The saturation level become reduced. So in presence of carbon dioxide, the affinity of hemoglobin with the oxygen is very much reduced. From 100%, it become reduced to 70 or 60, so that the curve moves to the right. This is called Bohr effect. So because of the carbon dioxide, affinity is reduced, and because of the reduction in affinity, the curve is moving to the right. Meaning, now, though oxygen is having the same partial pressure, the saturation has become reduced. Who reduced it? Carbon dioxide reduced. So, the effect of carbon dioxide in reducing the affinity of oxygen with hemoglobin when oxyhemoglobin or blood reaches the tissues is called Bohr effect. We can represent the Bohr effect like this. The curve is moving to the right. What is the reason for that? Carbon dioxide is more. So that carbon dioxide partial pressure high in tissues causes a reduction in the affinity of hemoglobin with oxygen. ഓക്സിജനും ഹീമോഗ്ലോബിനും തമ്മിലുള്ള ബന്ധം കുറച്ചു. ഹീമോഗ്ലോബിൻ ഇപ്പോൾ വളരെ കുറച്ച് ഓക്സിജനുമായിട്ടുമേ കൂടിച്ചേരുന്നുള്ളൂ. അപ്പോൾ 100% ഉണ്ടായിരുന്നത് ഇപ്പോൾ ഒരു 70% 60%ആയിട്ട് കുറയും. അപ്പോൾ ബാക്കിയുള്ള ഓക്സിജൻ എന്ത് സംഭവിച്ചു? ദാറ്റ് ഓക്സിജൻ ഡിഫ്യൂസസ്. അങ്ങനെയാണ് ഓക്സിജൻ ടിഷ്യൂവിലെത്തുമ്പോൾ ഓക്സിഹീമോഗ്ലോബിനായി സ്പ്ലിറ്റ് ചെയ്ത് തിരികെ ഓക്സിജൻ ടിഷ്യൂവിലേക്ക് പോകുന്നത്. അപ്പോൾ അൽവിയോലിൽ വെച്ചിട്ട് അൽവിയോലിയുടെ പ്രഷറിൽ ഓക്സിഹീമോഗ്ലോബിനായി ടിഷ്യൂവിലെത്തുമ്പോൾ ടിഷ്യൂവിലുള്ള മാറ്റങ്ങൾ കാരണം, ടിഷ്യൂവിലുള്ള സാഹചര്യങ്ങൾ കാരണം affinity of oxygen and hemoglobin reduced. So, the associated oxygen level decreased. The remaining oxygen diffused into the tissues. ഇങ്ങനെയാണ് ടിഷ്യൂവിലെത്തുമ്പോൾ ഓക്സിഹീമോഗ്ലോബിൻ സ്പ്ലിറ്റ് ചെയ്തിട്ട് കുറച്ച് ഓക്സിജൻ അങ്ങോട്ട് കൊടുക്കും. മുഴുവൻ ഓക്സിജനും കൊടുത്തിട്ടില്ല. സാധാരണഗതിയിൽ 1 gram of hemoglobin, ഇത് എഴുതിയെടുക്കുക കേട്ടോ. 1 gram of hemoglobin can combine with 1.34 ml of hemoglobin. 1 gram of hemoglobin can combine with 1.34 ml of oxygen. നമ്മുടെ ബ്ലഡിൽ 100 ml of blood, 100 ml of blood will have an average of 15 gram of hemoglobin. ആ 15 gram of hemoglobin അപ്പോൾ എത്ര ഓക്സിജനെ കൊണ്ടുപോകാൻ പറ്റും? 1.34 into 15, that is almost 19.4 ml. You round out to 20 ml. അപ്പോൾ 20 ml ഓക്സിജനെ കൊണ്ടുപോകാനുള്ള കഴിവാണ് 100 ml ഓഫ് ബ്ലഡിന് ഉള്ളത്. ഓക്കെ, നമുക്ക് അത് അവിടെ അവസാനിപ്പിക്കാം. ബാക്കി അടുത്ത ക്ലാസ്സിൽ. എനി ഡൗട്ട്? Okay, sir. Okay. So we will meet next time. Okay. Thank you and good night.